All right, everyone, this is Miss Slade, and we're going to be talking about non-Mendelian genetics. Yes, it's Miss Slade this time, not Miss Crowder. I hope you can adjust. Just to give you a little heads up on how I'm going to do this, I have everything pre-written, so I'm just going to be talking about it, and I'm probably going to talk pretty fast. So please use the pause button, please use the rewind button, and go through things and make sure you have everything that you need, and also pause. I will give you heads ups on good times to do so. Pause, and then make sure you have down all of the notes per section. So, what are we talking about? We're talking about non-Mendelian genetics. So far, this unit, we've been talking about Mendelian genetics, which is when there's one dominant allele and one recessive allele. Back in the 1800s, when Mendel first figured out dominant and recessive, he did it using pea plants. And these pea plants are a lot more simple than you or me. And so in reality, non-Mendelian genetics is much more common for what we see. There's very few of our traits that are just Mendelian, where you just have one dominant and one recessive. We're going to look at 4 different types, and just know that when we're actually looking at animals, often these different types are combined. So it gets pretty complex and confusing. But what we're going to look at right now is much more simple. So the first one that we're going to look at is incomplete dominance. So incomplete dominance is when there's 3 phenotypes, 3 genotypes, and 3 phenotypes. So what this means is that you have a particular allele that's not completely dominant over the other one. And so the resulting heterozygous result is its own separate phenotype, and it's usually somewhere in the middle. It's a blend between the 2 different traits. Sometimes it's a little unexpected, but it is somewhere in the middle between those 2 traits. So we could look at this from an example of flowers. Say we have a bunch of red flowers and we have a bunch of white flowers. If we were to cross those flowers and we were to end up with pink flowers, we would know that we were dealing with incomplete dominance, because pink is basically what happens when you mix red and white. To show this, we show it a little bit different. So we use a capital letter to represent the gene, but because we have incomplete dominance, we don't have a truly dominant and a truly recessive. We're just going to use different letters to note the 2 different alleles. So we've got C and then a superscript as R for red, and we've got a C and a superscript as for W as white. And then the heterozygous has both. Just like you would look at with Mendelian genetics, it's just noted a little bit differently. If you're going to look at a different example, say you had a plant or an animal that was this simple. I'm going to let you know humans are not this simple on this one. But say tall is incompletely dominant over short. If somebody was heterozygous, they would be medium. They would be somewhere in the middle between the 2. All right. Pause. Make sure you've got all of these notes down. This is on page 106 in your notebook. We're going to move on to the next section. So, codominance. Codominance is very similar to incomplete dominance, but it is not the same thing at all, and they can get easily confused for each other. So incomplete dominance is when the resulting phenotype for heterozygous is somewhere in the middle between the 2. It's some sort of combination, like red and white making pink. Codominance, on the other hand, is when both traits are fully dominant, so both alleles are fully dominant, so the resulting phenotype shows the traits for both alleles. What this means is, for example, chickens are a great example of this. Some chickens are homozygous for black feathers, and they only show black feathers. Some chickens are homozygous for white feathers, so they only show white feathers. However, if they're heterozygous, they have some black feathers and some white feathers. They've got a mix. Both traits are dominant, both traits are completely shown, they're not blended together. So you've got some black feathers and some white feathers. It's not the same as incomplete dominance because you're fully expressing the trait. You're just expressing 2. All right, pause again. We're going to go on to the next section, so make sure you've got this all written down, and we're going to move over to page 107. So take a moment, pause, write everything down, and rewind if you need to re-listen to that so that you understand. All right. Page 107, multiple alleles. So, sometimes there are more than 2 alleles for a particular gene. Sometimes there are multiple. A great example of this would be from your Drake game, where tails, there's actually 3 different types. You could have short, long, or kinky. They're all different alleles for the same gene. A real life example would be blood type. So, blood type, if you're looking at just the letters, because there's also a positive and negative component, which we're not going to talk about quite yet, if you're looking at just the letters, there's 4 different blood types that you can have. You can have B, O, A, and AB. And that might not mean a lot to you right now, but what it actually is showing is that there's 3 different alleles that you can have. So there's A, B, and O. O is recessive. A and B are codominant. And so how that actually works on a blood cell. On a blood cell, there are different markers on the outside of it. If you remember way back when we were talking in our cancer unit about HLA matching, it's very similar. There are these markers on the outside of the cell that can tell what basically if it's a friendly cell or if it's a foreign cell. And so, for example, if you've got type AB blood, you could have both A markers and B markers. If you just have type A blood, you're only going to have A markers. If you have type B blood, you're only going to have B markers. But because it's codominant, when you have AB, you have both. You'll have A and B. O doesn't have any markers, and it's recessive. So O, if you have type O blood, that means you have none of those markers on your blood. If you were to cross a parent that has heterozygous type B blood and then heterozygous type A blood, and they were to have a kid, you could actually have all 4 different phenotypes as a possible result. So they could end up with AB, they could end up with just B, they could end up with just A, or they could end up with O. So it gets much more complicated than some of the more simple Mendelian crosses that we were doing before. Just to add to this complication, there's a different gene that also adds markers to our blood, and that's the positive and negative. And so if you have the positive, you have those markers as well on your blood, which is why you'll hear that somebody is AB positive. That means they have the A markers, they have the B markers, and they have the positive markers. Interesting to note, some of you have heard that O negative is the universal donor, and that's because they don't have any markers whatsoever on their blood. They are negative, so they don't have the positive markers, and because they're O, they don't have the A or the B markers on there. So that means there's nothing on those cells that another human body would recognize as foreign, so they can donate their blood to anyone. Unfortunately, it also means that they can only accept blood from other people that are O negative, because any other type of blood is going to have a marker on it that they don't have, and their bodies will recognize it as foreign. On the flip hand, if you have AB positive blood, you are a universal receiver. You can accept blood from anyone because you have all 3 markers on your blood. So even if it's from somebody that only has if they're type A negative, you can still accept from them because they have the A markers, and it doesn't matter that they don't have the B or the positive. So if you got AB positive, you're set. You can receive from anyone. However, you can donate to very few people. You can only donate to people that have AB positive blood. So just to keep in mind, the positive and negative is an additional gene. And when we're talking about the letters, there are 4 different phenotypes for only 3 alleles. All right. Pause. Make sure you have this all written down. Rewind if you need to. We're going to go on to the last section, which is sex-linked traits. So you found this with the no spike on the drakes, and it was very frustrating for some of you. This is when the gene for the trait is found on the X chromosome. So this means when that gene is recessive, it is much more likely to be expressed in people that are genetically male. An example of this would be hemophilia. If somebody has normal blood, the allele for that is dominant. So they only need to have one X chromosome that has that dominant allele, and they're fine. They're going to have normal blood. However, if you only have one X chromosome because you've got an X and a Y, it means that you only have one chance on that allele. So if you have the recessive one, there's no dominant to mask it. So it's going to show. How this works is that if you were going to, if somebody that had totally normal blood, a father that had totally normal blood, had a baby with somebody that was heterozygous, so they also had totally normal blood, but they are carrying the allele, that recessive allele. If they have a child that is genetically male, then there's a 50% chance that that child will have hemophilia. If they have a child that's genetically female, there is 0% chance. And overall, it's 25%. Examples of this would be color blindness. It's much more common with people that are genetically male because it is recessive and it's on the X chromosome. And as well, male pattern baldness is another example. Where you will see that it is much more common, and that's why it's called male pattern baldness, because it's recessive and it's on the X chromosome. All right, that is the last of these 4 different patterns. If you have any questions, please reach out to me or Ms. Crowder. And if you need to go back and review any of this, rewind, I highly recommend it, or pause to just make sure that you have all of the notes in your notebook. All right, thank you, everyone.